Thanks, folks who have dialed in so far. There may be some more dialing in momentarily. I hope everyone is doing well and staying safe. We're really grateful to have Dr. Harley Newton give a Zoom presentation today. But before I introduce Harley, I just want a couple quick updates. We're kind of excited to share that a new paper on our juvenile white shark tagging research has just been accepted and will be published within a few weeks. So stay tuned for more details on that. We also have some exciting updates for future programs, Zoom, and hopefully some in-person programs at SOFO coming up, and also some more continuing research, hopefully this season, with some exciting new collaborations we have going. So please stay tuned and follow South Natural History Museum and the SOFO social media handles and stuff to keep updated on that and all those updates. So and thanks to SOFO for hosting this. So we'll get to Dr. Harley Newton. Harley has been a fantastic collaborator for the last several years with our white shark research and studying other species. Initially, when we started working together, she was based at the New York Aquarium and the Wildlife Conservation Society. She's now working for Disney down in Florida, where it's a little bit warmer, less snow. You've heard some of you who have attended some of our programs before. A lot of our focus has been on tagging and the field work that we do with the sharks off New York here. But Harley's going to talk about something completely different, which will be exciting, and working on the stress and health assessments of the sharks. So Harley is a veterinary pathologist. She's one of the main people drawing blood from most of the sharks that we tag, and turns out you can learn a lot of things from shark blood work. It's certainly out of my realm of expertise, so having Harley's expertise with our research team has been really insightful and helpful, and it's been a fantastic collaboration, and she's done some really great work. So we're really happy to have her this morning to share some progress on what all that analysis is showing. If you have questions along the way, please use the chat box, and we'll try to reserve them for the end of the presentation. But if you have questions, please just put them in the chat and we'll get to them towards the end. So with that, I will turn it over to Harley and she can put up her presentation. But thanks again, Harley, for being here, and looking forward to the talk.
Awesome. Hopefully this is showing up correctly. I just want to check.
Looks good on my end.
Okie doke. Great. Well, thank you everyone for being here on this Saturday morning. It is actually very nice and warm here in Florida, but raining. So if you hear lots of thunder and such, that's what that is. So my presentation today really is just going to be an overview of the type of health and stress assessment work that we've been doing in Northwest Atlantic white sharks over the last, gosh, I think it's been 5-6 years now that I've been associated with this team. And I just wanted to start with a thank you because, I was already doing some shark research in the mid-Atlantic when I was at the New York Aquarium. It was primarily sand tiger sharks, and we'd been trying to expand from there and doing some other species, but it really wasn't until I got the opportunity to work with Toby and Greg and Matt Ajamian that I really got to expand my horizons, be exposed to a lot more species, and really start taking a deep dive into some of the questions that meant the most to me as a veterinarian who's interested in elasmobranch health and conservation. So globally, so globally, sharks are landed for their meat, their fins, liver, and oil, and they are significantly in decline, with over 71% decline of sharks over the last 50 years, and a steady 18% decrease each decade, despite the regulations that are in place. Now, fortunately, white sharks have shown some regional recovery that's been aided by retention bans, but the reality is that sharks are really at high risk, despite the regulations that we have in place. And these numbers actually come from a publication that only came out 2 weeks ago in Nature. So health is an important conservation tool. And what I tend to practice is what I call conservation medicine. It's a interdisciplinary field that studies the relationships between human, animal, and environmental health. And it basically recognizes that animals are really critical sentinels to environmental disturbance, which obviously makes sense. We're all interrelated and we share the same ecosystem. So the same risks to health are present for all of us. It's really important to understand the impact of acute and chronic human stresses on wildlife populations, and also the role that emerging diseases may play in population declines. And that's largely because a lot of the policies that we put in place to address things like fisheries, assume that we have healthy populations. And if we don't look at the whole picture, we don't know the health status of these populations, we may not see the kind of response to the protections that we put in place. There may also be other risks out there, such as disease or other stressors that we don't know about. So it's really critical to use health as a tool for conservation. So what is health? It's most simply defined as being a state of physical and psychological well-being and of productivity, including reproduction. So basically at its core, it's the ability of animals to adapt and self-manage. So feed themselves, maintain body weight, demonstrate normal behavior, reproduce normally. Those are the kinds of things that we consider as healthy animals. Population health, so at looking at health from a population level, not just an individual animal level, gives us some, again, the critical insight into the physiologic effects that the environment might be having on animals, what humans stressors might be in place, any underlying disease issues or other dynamics. The challenge with sharks, though, is that we just don't have a lot of baselines. So some of the thrust of our research is really just creating those baselines of what is normal health, what is that in a wild shark, a wild white shark, so that we have something to compare to. And it's critical to collect that information now because the ocean is changing and it's changing really rapidly. So what's normal now may not be normal in the future. So it's important to collect a lot of the information I talk about today now, so that we have it for future studies. Stress for me is kind of a buzzword because I think it means a lot of different things to a lot of different people. But basically, when I'm looking at stress, I'm looking at actual physiologic impacts that an event has on the animal and what their response is to an event. Fisheries captures impact the survival of sharks after the release. So it's important for us to understand and be able to predict what sharks can handle certain handling and what sharks can't, what animals are going to do well after the release and what ones aren't. But the reality is that, again, similar to these health indices that I talked about and will talk about, we really are still learning a lot about exactly what a stress response is in an elasmobranch bank, because it is different from mammals and the things that we're used to. And then how we can use it as a predictor of how well the animals will do after they're captured. So the goal of the health research that we've been working on together is to perform health assessments and establish reference ranges for white sharks, to identify any diagnostic trends or emerging population issues along the coast, and then also document the stress response we're seeing, and then try and correlate that to an overall picture of health of the animals. And additionally, use that information potentially to inform ocean and other fisheries policy. So when we talk about health assessments, this is no different than when you go to the doctor. It's, looking at you to see that you're in good condition. And in our case, it's also collecting blood samples and looking at those to look at organ health, signs of inflammation, and other indicators. So in marine species, it's really challenging to rely on outward physical or behavioral changes to infer health, as they're very difficult to observe. I think, all of us know if our dog or cat is sick because they're right there and you can see them and you can see that their behavior changes. obviously the oceans are vast, and most of these animals that we see, we're only interacting with for maybe tops 14, 15 minutes. So it's really challenging just to look at an animal and be like, oh yes, that's definitely a healthy animal. And that's why the blood samples that we've been collecting and analyzing is so critical for trying to establish, you know, metrics, trying to establish actual health parameters that we can look at and compare. And blood chemistry and blood work is really an important component of monitoring health in all wild populations, humans. To date, we've sampled 68 white sharks from 3 different age classes, and that includes 26 young of year juvenile white sharks in the New York Bite with Toby and Greg and your team. And the rest of the animals have been sampled over the years on expeditions with OCEARCH, and those tend to be the subadult and adult age classes. And the types of testing that we're doing is really not that different than what you or your pet would have when you go to the doctor. So we're looking at things like hematocrit or packed cell volume. That's basically just a ratio of the red blood cells to the plasma that gives you a sense of what your red cell count is and whether it's normal, whether you're able to carry oxygen normally. We're doing complete blood and differential counts. I think maybe if you've heard of having your CBC done, this is looking for the percentage of different cell types are there, particularly white blood cells, cells that respond to inflammation. So we're looking at what cell types are present and what the ratios are of those cells to try and establish health. Chemistry panels, I think most people are sort of passingly familiar with. This gives you information on organ health. So it gives you like your liver enzymes, your kidney values, it'll give you electrolytes, and it'll give you kind of a snapshot picture of organ health. And then total protein and electrophoresis, this is useful and used in humans and your pets as well in looking for markers of inflammation. So this sort of top group of parameters is not unusual to perform. The bottom set of parameters are really something that's a little more specialized to elasmobranch research. And some more specialized markers of inflammation. So acute phase proteins are just proteins that are made in the liver in response to inflammation. For the purposes of shark research, sometimes it's referred to as a tertiary stress response, in that these are things that tend to be impacted by sort of environmental stressors or other stressors or illness. 1-alpha hydroxycorticosterone is the primary stress hormone in elasmobranchs. They don't use cortisol. And for the last, 30 years, it's been a challenge to have any sort of assessment of this possible at all. And I'm fortunate in that the endocrinology lab here at Disney did develop an assay that can actually measure the level of 1-alpha hydroxycorticosterone in sharks. Prior to that, we couldn't look at this primary stress hormone. Traditionally, what's been used in sharks is looking at their blood gases, their lactate, and a few other analytes to see how those change with exertion. So these bottom group of tests are what we are typically looking at when we're trying to assess stress in these animals. So without further ado, I really just wanted to share some preliminary findings in these cases now that I've talked you through kind of what we've been doing over the last few years. So I'll start with the cell counts, which may not seem all that interesting, but I get really excited by them. So the only paper that existed prior to this that had any information on what a normal packed cell volume, or basically, a red blood cell type count, was in white sharks came from a group of 5 deceased animals that were assessed in 1986. And that showed a hematocrit of 36. For reference, in people, we tend to be sort of similar. We're between like 35 and 45 or up to 49. And what our health study has showed us is that, yeah, that's a pretty accurate number. So of the 68 animals we've looked at, we're coming up with on average a hematocrit of 36 and very little variation among all the age classes. So across all the age classes, this is a pretty standard number. More excitingly, we've been able to define the white blood cell characteristics. And I say more excitingly because elasmobranchs present kind of a wide range of white blood cells that all look very different. So one of the challenges we always run into when we first make a smear or a slide is even figuring out what their white blood cells are and what their function is. So over the past few years, what we've been able to do is identify each cell type. And we've actually been doing some special stains because these little pink granules that are in many of these cells have other characteristics that tell you what the function of the cell is and what it's there to react to when there's an inflammatory process. We've also done counts of these white blood cells and also proportions to each other. And the interesting thing that we've found in the Northwest Atlantic white sharks is there's no variation among the age classes, but we are seeing some variation in our group of animals versus the only other publication that exists, which was in South African white sharks. Our sharks are probably have a white blood cell count that's twice what they were seeing in South Africa. And I'm not sure whether that's just based on the method that they use to assess them. We're doing actual counts where we actually put the blood into a chamber and count the individual cells, and they were doing slide estimates, or whether there is an actual difference. One of the other really interesting things to come out of this work is, like I said, ratios of the different cell types. So these types of cells, granulocytes and neutrophils, tend to be the ones that come in and respond when you have a new inflammatory process, something that's causing an illness, versus your lymphocytes, which tend to be more predominant in shark species. So what we've been seeing in most sharks is that if animals have active inflammation, you'll see more of these compared to these, and you'll have a shift in the ratio. And that's important because one of the things that we've seen is that when we go through an assessment of all the animals we've sampled so far, there was no difference between age classes, gender, any of those things. But definitely where we sampled the animals and when we sampled the animals, we were seeing a different ratio of those granulocytes to lymphocytes. And what does that mean? This higher number means there's more granulocytes than lymphocytes. So it suggests that animals in this region during this season might have a little bit higher of an inflammatory response going on. And that was interesting because we also saw some unique changes in these animals that really have only been reported in humans. And these are called extracellular trap formation, which are these little projections that stick out, and they're there to specifically interact with infectious agents. They can happen with autoimmune diseases in humans, but they're also there in response to inflammation. So that's two things that we saw that would suggest that animals in this region during this season were showing some sort of inflammatory response that's not present in other animals in other regions in other seasons. And then the final sort of marker of inflammation, I told you about those acute phase proteins, those proteins that are made in the liver in response to sort of external stressors or active inflammation. And it was kind of the same story with these, which is, again, particularly C-reactive protein, which is something that elevates pretty quickly when you're having an immune response. We were seeing, again, that same region, that same season, much higher values. So that would, again, suggest an inflammatory response. And I have the normals down here. That doesn't really matter, but I think you can see that if the mean CRP is 74 in these animals in this region and season, and animals in sort of Massachusetts or New York are only 0.1 and 13, there's definitely something interesting going on in this population during that time. Now, we can't say that animals there, we can't say what that is. It's really challenging because these things could be elevated due to infection, exposure to toxins, exposure to other environmental stressors like humans, but they could also be to social stressors. So if there was, a lot more sharks in an area because they're breeding there or feeding there or other interactions, those all have to be taken into account. So it's super interesting that this region and season are showing this change, but I'd be getting ahead of myself if I told you that I thought we know what it means yet. So the next exciting thing that's come out of all of this work is just the very beginnings of having chemistry reference intervals. So we all, sort of take for granted that when we go to the doctor and he gets our blood work back or she gets our blood work back, they know what those numbers mean. And they know what those numbers mean because they have reference intervals. And human reference intervals are super specific. I mean, they're down to like what age you are, what country you live in, they're all very specific. In sharks, I'm lucky if I can come up with an animal that's pseudo-related or has a similar lifestyle. So having these types of reference ranges to set a baseline for normal now so that we can evaluate animals in the future as the ocean changes is just super critical. And we've reached a point now where for certain analytes, and mostly these are just electrolytes, where we have we have enough to have a statistically significant reference interval for those. So that's pretty exciting to have that. We obviously have, there's 29 different analytes that we look on that panel, including liver enzymes and other kidney values and such. Typically, once you get to 60 animals, you should have enough, but we're at the stage now where we probably need a few more animals in order to make some of those statistically significant and be able to come up with similar ranges. But this was really exciting to finally have. So as far as stress physiology goes, traditionally, what has been used for evaluating stress is looking at changes in blood gases and changes in muscle enzymes and a few other analytes that are basically induced by stress or induced by exertion during capture. So that largely is just because of this shift that you go through when you exert yourself into what's known as anaerobic metabolism. So you have, muscle lactate values, which I think might be familiar, that become elevated, and then your blood gases change. You'll have higher CO2, less O2, those types of changes. This is what's traditionally been used in elasmobranchs, banks, but what we've been seeing over time is that this type of analysis may not be able to predict which animals are so significantly stressed by the handling event that they don't do well afterwards. There's certain species that can tolerate really high, big shifts in these values, and then there's certain species that can't. So that's why we've been on the lookout for maybe other markers that might be a little better at doing this. But in the meantime, what we've seen is a few significant differences that were expected. So animals that had kind of a longer time from the time they were hooked to capture or had a longer recovery time tended to have a lower blood pH. So again, as you exert yourself, your blood pH will decrease. So that was not unexpected. And again, muscle lactate levels, these lactate levels were higher if the time to capture was longer and the recovery time was longer. One interesting thing is we did tend to see lower lactate values in the young of year animals. And that was a pretty important question that I think we've been focusing on regionally, knowing that that the Mid-Atlantic was a nursery site for white sharks. And that was, how vulnerable is this life stage to fisheries capture? So if these animals were showing signs of significant stress or big physiological disturbances that might suggest that they aren't going to do well afterwards, that this is an important life stage to have survive and grow to adulthood. So it's important to know. So it was interesting to see that these animals had lower lactates, but the reality is that these were smaller animals. They took less time to bring in to sample. So that may be one of the significant effects that we're seeing, just that shorter handling time. The other thing that we were seeing across the board is that the blood gases in these animals were stable during handling. And from my perspective as a veterinarian, that was important because we were handling these animals a lot of different ways. Certainly when I'm out with the crew from SOFO, we're doing all these animals boat side, and they may be ventilated or not. And the blood gases were relatively stable in those animals. And then when I'm working with OCEARCH, those animals tend to be put on deck and artificially ventilated. And again, blood gases were stable during handling. So those were important things to look at with these animals, just as far as the handling that we were doing from a research perspective. So this is relatively new information that I just presented at AES this year. And that was looking at, as I explained to you, this response, which is 1-alpha-hydroxycorticosterone. And that's so basically the shark version of cortisol. So when you're stressed, your adrenal glands will release this hormone and induce a response. And what we're looking for is how extreme is that response when these animals are undergoing handling. And what's interesting in white sharks is that it's really low. And so this was our lower limit of detection. These were all of the animals that we tested. And basically, all of these animals fell between, 1 and 2 or up to 3 nanograms per mil. This is an extremely low level. So we've looked at a lot of different species and a lot of different situations, both in managed care and aquariums, as well as animals that were handled from different methods of capture, like long line. And what we see is that other sharks that are experiencing illness or other prolonged stressors have much higher responses, well up into the 20s. So this can be a real predictive indicator. So it's super interesting that these animals undergoing similar kind of fisheries stressors or research capture stresses really show very little 1-alpha response. We did compare across all the different age groups, the sex of the animals, how long they were being handled, captured method, wasn't any differences. There were no significant correlations with those secondary stress indicators, so there wasn't a correlation with the lactate values, the blood gas values. But what this really showed us is just that we're really not 100% sure what the range of the response actually is in white sharks. Are they really using this hormone, this 1-alpha-hydroxycorticosterone, as their primary stress hormone? They should be, but we don't really know. And what came out of this research is that we probably need some more kind of extremes and different methods of capture. Most of the methods of capture that we have are, in the grand scheme of things, pretty short. Certainly rod and reel fishing, and then the young of year sharks, really short time to get those animals to the boat. Some of the larger animals might take a little more time, but in the grand scheme of things, we may just not be handling them long enough to really see what their full capability of response is. Just checking time because I tend to talk too much. So future directions, we have a number of additional questions that have come out of this research that I think are important, and a lot of them come from just wanting to better understand the physiology of these animals from a stress component, as well as just general physiology of these animals during handling. So one of the things that we've started doing is using inline accelerometers on the lines that are used to capture the animals. So in the larger animals, these tend to be drum lines. And what we're looking for is the forces involved, the forces that these animals exert when they actually get hooked and up to the time of bringing them to the boat. So really being able to correlate all those changes I talked about in the stress values. So like the 1-alpha levels, the lactate levels, the blood gas levels, understanding how that correlates to how much exertion the animal actually made during that process. Some sharks are very relaxed about the process of being brought to a boat, other ones are not. And if we're not accounting for that, we're not capturing that, then we may not really understand what disturbances in those values really mean. So this is an example of a very large female animal that we sampled on OCEARCH that was caught on a drum line. And you can see here, I mean, the point at which this animal is brought up and the fight, and then just these are all just vectors of force showing the exertion between the animal during the capture time. So this is just one. We've done a very small number of these, but something that we're looking forward to doing in the future. The other thing that we really want to understand better is heart physiology, and this is a relatively new collaboration that we're working on now with University of California, University of California, San Diego, and Carolina Coastal University. And that's just getting a better sense of the actual physiology of the heart, how the heart's actually functioning when these animals are being handled under different methods of handling. So I have, hopefully for you, we're doing this largely using cardiac ultrasound. So this is a white shark heart. You can see the ventricle here, the valve here, and this is the outflow tract here. And this is just in the animal's heart beating. And our hope, is that we'll be able to really look at actual heart function in these animals while they're being handled, to understand what their contractility is, what the velocity of blood flow is, and maybe use some of our other values to estimate oxygenation. And I guess I talked a little bit faster than I thought I would, but it does leave a lot of room for questions. We still have a lot of questions about these animals. I've really just given you a snapshot of some of the early information that we have. I think a lot of this information is ultimately going to be pretty critical to conservation efforts because, as I said to you, a lot of conservation efforts are aimed at stopping sort of, physical disturbances to these animals' environment. If you don't take into account whether these are healthy populations to start with, you may not ever see whether these animals you may not see the response that you're expecting to see is what it comes down to. So you may not see a population increase, and it may not be that what you have put in place to protect them didn't work. It may be that it's not a healthy population due to other reasons. So that's one of the reasons that I feel really passionate about doing shark research, is because I feel like I can fill in this extra gap that a lot of people can't, essentially, and really contribute to conservation in my own way as a veterinarian, which is somewhat unique. And with that, I guess we're going to have more time for questions than I anticipated. I didn't mean to talk that fast.
All right, that's great, Harley. Thank you so much. Really, really insightful presentation. I guess if folks have questions, if they want to put them in the chat box, I can try to moderate a little bit. We have one so far from Paul. Is the data you presented published yet?
Most of it's been shared at meetings, but some of it is in the process of publication. The challenge that we're having with a lot of the blood work values, again, is just animal numbers. We just don't have enough for statistical significance, but I think we're probably getting there with some of the preliminary secondary stress indicators. So I think we're almost there.
Great. I have a question. I guess, how do you see, do you think there are ways, just in kind of general terms, we can connect, obviously our focus, my focus, and SOFO's focus is kind of on the tagging and the movements and the behavior after they're released. Do you think there's maybe some novel ways to sort of incorporate your stress assessments with the post-release behavior? And you think we can, there might be ways to sort of connect the dots between our movement data, behavior data, and the blood work?
Yeah, I mean, some of the tags you use are specialized enough, like dive profiles and some of the other information that you guys collect. I think it's going to be really critical in understanding some of these stress values. So the challenge is, I was hoping that the 1-alpha hydroxycorticosterone levels would be much more telling and higher, and we'd be able to do a little more with those. But I think looking at the blood gases, looking at the lactates, and then looking at the dive profiles and seeing how quickly these animals return to what we would consider a normal pattern would certainly be one way that would be useful. And then for me, the challenge with being a veterinarian who's collecting my little subset of data is, you need all that other natural history data to interpret it. So I can say all this interesting stuff about having these inflammatory profiles in the southeast in the winter, but without having a group of people who can tell me, well, who's down there and what are they doing and where are they and what might they be exposed to, like that's going to be another critical piece for better understanding that one little blip of information.
Great. Any other questions coming in? Here we go from Greg. Do you have a list of hormones that may give you the response you're looking for? Do you, for sure? Yeah. Do we think we've covered our bases? Are we looking in the right places?
Yeah. So the nice thing with the laboratory that I work with is we can do some other things. So there are plans to look at some other markers, and then also looking at norepinephrine, epinephrine, and then also looking for other metabolites of cortisol and hydroxycorticosterone. You know, we're not just jumping to the assumption that because all sharks use that, that's the only thing out there. But I think for us, you know, we just probably need samples from animals that have undergone different stressors than what I currently have, which is always the challenge with research. You know, you have your methodology and you're trying to keep the handling as quick and easy as possible. And for something like that, sometimes you need something different. So yeah, I'm not sure when we're going to get to the norepinephrine and epinephrine. I'm kind of excited to see those whenever that gets available.
Would you be able to comment a little bit about your experience with some of the sort of more biological pathogens and parasite infections and stuff that you've experienced in New York? We've had a couple sort of mass mortality events, dogfish and things washing up. There's been some occasionally the spring and summer we get white sharks and thresher sharks, other species that wash up dead on Long Island beaches. Could you comment a little bit on some of your research and data collected on some of those strandings?
Yeah, you know, traditionally, certainly for sharks, disease is just not one of those things that's often considered to be something that's impacting populations because the focus is so much on human interaction and fisheries. So we've had a few events, and typically what happens is when there are mortality events, the first thing that I get told, particularly if it's a small shark species, is that it was probably a fishing trawler. And I think that was some of the big surprises, is that a lot of those mortality events are not investigated. So those animals are not examined. They don't go through a process of autopsy, or in animals, we call those necropsies, where you collect tissues and really ask the questions of like, were these normal animals or were they not normal animals? So one of the things that came out of that is certainly around Labor Day of 2018, there was a mass mortality event of smooth dogfish off of Coney Island. And the assumption was that it was, you know, just again, fisheries related. But when we looked at those animals, they actually had a parasite infection that was actually infecting their brain, as well as systemic. And it's a parasite that's been identified on the Pacific coast as causing significant mortalities. So it was the first time it's been identified on this coast, and it, for probably a 3-week period of time, caused significant deaths in that population. There's some bacterial infections that have been recognized in juvenile salmon sharks on the Pacific coast that are important, again, causing brain infection and causing mortalities. And then certainly along Long Island, I do still collaborate with the New York State DEC. And when there are shark strandings that come in along the coast, I used to go and do those evaluations. They now go and collect tissues and do evaluations. And we're still trying to investigate the hows and whys of why those animals come in. So we've had a handful of thresher sharks, a handful of white sharks, and it's been really valuable, both just from a life history perspective, because when you do those exams, you can see what they were eating, what their body condition was, and then also looking just for signs of health or inflammation or sickness in their tissues. So literally, 3 days ago, I got my box of tissues from the last season, which I'm going to have to look at. So yeah, it's investigating natural mortalities or animals that are found dead is just critically important if you're going to understand what the real pressures are for animals in your region.
So it looks like Greg would also like to know, is there any additional data we could be collecting that would help? You know, obviously we were happy when we have you along on the boat with us, sampling these sharks with us. It's a lot of fun. But especially over the last year, it's been tougher to travel and
Yeah.
get everyone on the boat together, get the A team together. So is there any additional data we could be collecting when you're not there that might help?
You know, I think, you know, we're kind of entering that phase where there's definitely some tools that I've been using in the larger animals that I'd love to start using in the young of year to try and better capture what's going on with them. So I mean, the line accelerometers would be great to send you guys some of those. And I now may or may not own a bunch of them. It's the problem with grant funding is sometimes you just get impatient. You're like, well, they're not that expensive. I can own a few. So that's one thing that I've been thinking about. And, you know, the blood work is always critical. You know, I would love to come and get some cardiac ultrasound on these little guys too. I'm really excited about that. I have so many questions about how these animals really are being handled by all the different methods that I wind up using depending on who I'm working with, and I think that could also be impacting some of our blood values. So I think that could be really interesting to do too. Now I can't send you an ultrasound to use. You're gonna have to take me with you.
All right, definitely we can work on that. That's one thing we didn't really talk about yet was you know, how white sharks are regional endotherms. They're partially warm-blooded. That has a, you know, an effect. Temperature obviously isn't a big driver in you know, thermal physiology and stress physiology, the stress response. Is there, I don't know, maybe you could comment just a little bit on what makes white sharks special in that regard, and how that ties in with their health and stress tolerance.
Yeah, the whole regional endotherm component has been an interesting one, and there hasn't been a lot of publications about their response to about their stress responses. There have been a few papers in MACOs that show that regional endotherms may be able to more quickly swap from anaerobic metabolism, which they enter when they're exerting themselves, back to aerobic metabolism. And because of that, it may mean that they can survive bigger physiologic shifts in the things that traditionally we would think are not survivable. So super high lactate levels, big changes in blood pH, those types of things, they may be able to uh, shift back into aerobic metabolism and correct those fast enough that they don't have the type of mortality you would see in another species. So I think that is definitely one big question that we're going to have to answer.
That's great. Yeah, so there is some evidence that being warm-blooded is kind of a benefit in terms of handling stress potentially.
Yeah, for sure. And I think that's where some of our questions come from with the hormone levels being so low as well. Like, why are they just not entering the type of stress response that we see in so many other animals?
Right, that's great. Last call for any chat questions for Dr. Harley. Okay, well, I guess we can wrap it up. Oh, Walt jumps in. All right, thanks, Walt. As far as
Thanks
collecting
for your
blood,
picture, Walt.
as far as collecting blood samples, is there any effort being made to develop an easy and reliable method for collecting blood samples?
Well, I will tell you that
I guess he's meaning for clumsy, you know, taggers like us. Is there an easy
yeah.
way to poke a shark and get some blood?
So the challenge is getting the blood is typically not a problem. I feel like I can show a lot of people how to do it. It's what we call a blind stick, you know, so like when you go to the doctor can see your blood vessel. For sharks, the blood vessels run in this little canal. Well, the easiest access point to blood vessels are an artery and a vein that run in a canal underneath their spinal column. So that's what we're actually finding and poking through. And it's, like I said, it's what we call a blind stick. So basically I take a needle, I put it where it should be, and then I feel around for that, and then I sort of just feel in with the needle. I can typically teach people how to do that. The challenge is the processing. And I think that's the big thing, is that for all of these analytes to be valuable, the blood has to be separated very quickly. It has to be chilled very quickly. It has to be frozen very quickly. And in the case of the blood gas analyzers, it has to be run like almost immediately. So that's always been the challenge, because otherwise, there's lots of blood work you can do that doesn't require that kind of stuff, but not the kind of work that I do,
Right.
unfortunately.
So, All right, so it's possible, but may need some more. We all may require some more training to do the full workup for you.
Yeah. I mean, I will tell you that there's been a lot more publications, particularly in fish, and then some smaller shark species of using some more approachable single test meters. So like the types of lactate meters that like marathon runners use when they're just self-assessing, like a lot of those have slowly over time become validated. So the device that I use to run blood gases and lactates is pretty expensive, but those are a little more approachable and can be validated. So you can look at other things like lactate, glucose, blood pH using those smaller meters.
All right, great. Yeah, Greg says, sounds like we're gonna need you back up here to cover all this stuff for us. So hopefully we'll get you back up soon.
Yeah, I hope so. I hope so.
So, all right. Well, if there's no other questions, thank you again so much, Harley, for doing this. This is really, really educational presentation, new information for me, and hopefully everyone else got something out of it too. But please, please continue to dial into SOFO social media. Follow us at #SOFOSharks with updates on the research and publications and other programs and things. So, but thanks everyone for attending. Stay well, and we hope to see you at the next program.
Awesome.
Thanks, Harley.
Thanks, everyone.